Hi everyone, so today we're going to be having a look at the biological approach in AS psychology for EdXL. And in particular, we're going to be starting to have a look at the central nervous system and also the effect of recreational drugs on transmission in the central nervous system. Okay, so let's start off by looking at what the central nervous system actually is. So the central nervous system in the body consists of the brain and the spinal cord together. Connecting the nerve systems is nerves throughout the whole of the body. Okay, some of these nerves help us to move, which are obviously motor neurons, and there are other neurons that are that send information towards the central nervous system, such as sensory neurons, as in what we see, what we hear, what we feel. Okay, they send obviously information back to the central nervous system. Now, the brain is made up of billions and billions of nerve cells called neurons. And neurons pass information from one to the next around the whole of the brain and throughout your body. They then communicate with the rest of the body through nerve cells in the nervous system and telling different parts of the body to do different things. Okay, so for example, you lifting your arm to pick up something, that's the brain's neurons sending information nerves down your arm and obviously telling you to contract your arm to be able to pick something up. And the role of this whole central nervous system is to process all new incoming information, which obviously comes through from the senses. And what it does is sends messages to the different parts of the body. The neurons, so remember the brain is obviously made up of billions and billions of these, which is the key part to making the central nervous system work. And neurons communicate with each other through synapses, okay, which is the gap between two neurons. And this allows the brain to process information such as thoughts and memories. The neurotransmitters, so neurotransmitters are chemicals which affect our behavior. And what neurotransmitters do is they travel across the synapse, which is a gap between two neurons. And then as it obviously reaches the other side of the neuron, the other neuron, it then attaches itself to the receptor sites on the other neuron, or the postsynaptic neuron. You may have learnt about this at GCSE science, the lock and the key mechanism. So it's exactly the same as that. The receptor sites on the next neuron are unique to each type of neurotransmitter. So examples of neurotransmitters are things like dopamine, serotonin. So they'll obviously all have their own unique shape. So only one particular type of neurotransmitter can fit into the receptor sites. And the whole process then converts chemical signals back into electrical nerve impulses. You also, for your exam, need to know the structure of a neuron. So you need to be able to label a neuron and also describe some of the main key parts. So if we have a look, let's start off with the dendrites. So here you can see the dendrites. Okay, these are these branch-like structures around here, the edges. These are all dendrites. And dendrites actually receive messages from other neurons in order to trigger something called an action potential, which we're going to be talking about later. Okay, the whole of this here is the cell body, and the cell body contains the nucleus and the mitochondria. So the nucleus obviously contains chromosomes, which contains all the genetic information, and then also the mitochondria are obviously used to provide energy for the whole of the neuron. Okay, so you've also got this long sort of tube, almost like, which is called the axon. So the axon itself actually transfers the information from the cell body towards the other end of the neuron, which is the axon terminals. So here you've got the axon terminals at the end, which is the end of the neuron. So when the electrical message comes through, it gets to the end of the axon terminal here, and then from there it passes on to another neuron. Okay, now the axon itself is actually covered in something called myelin sheath. Okay, the myelin sheath is like a fatty layer which surrounds the whole of the axon, and the whole aim of the myelin sheath is to speed up the rate of transmission. So the speed up the how quickly the electrical message comes in and transfers from one neuron to the next neuron. Okay, you've got Schwann cells that produce the myelin. So the myelin is the fatty layer which obviously coats the axon. And in between these here, you've got the node of Ranvier, which is here, these little tiny gaps. Okay, so the node of Ranvier are gaps in the myelin sheath. And again, what happens is they basically speed up the electrical conductivity. So what happens is electrical message will come in through the dendrites, it will go down the axon. But this time, because we've got myelin sheath here, instead of traveling like this, it will jump between the node of Ranvier. So the electrical message will jump like that. Okay, and that is a lot faster than traveling from that to that. Okay, so for your exam, do learn the different parts of the neuron, and also you need to know what their functions are as well. Okay, action potential. So I mentioned action potential earlier. The action potential is basically the way the nerve impulse passes down the axon onto the next neuron, and the way it almost stimulates the release of the neurotransmitter into the synapse. So neurons have two types of potential. We've got a resting potential and an action potential. The average resting potential of a neuron is about 70 millivolts. Okay. When a neuron receives a message from another neuron, there are two things that can happen really. Firstly, it can either cause an excitatory response. So what happens is the neuron becomes depolarized and reduces its charge. Okay, so it goes lower. Secondly, an inhibitory effect could happen, which increases the charge. Okay, so two things could happen, one of two things. If the neuron receives more excitatory messages than inhibitory, then an action potential is triggered or stimulated. Okay, so during an action potential, the potential of the neuron can reach minus 55 volts. Okay, so remember, if it's being excitatory, then an action potential will depolarize it, so the charge will reduce. Now, the speed of which the action potential occurs is actually obviously made faster by the myelin sheath. So remember, we looked at the diagram of the neuron earlier. The myelin sheath is what acts as an insulator, and it obviously speeds the rate at which the action potential can travel from one side of the neuron to the other side of the neuron. And remember, the nodes of Ranvier are the gaps between on the axon, where the ions flow in and out of the cells to regenerate action potential. So the gaps, the nodes of Ranvier, are sort of open for ions to flow in and out. And the whole point of ions flowing in and out is to regenerate that action potential, okay? To get that information from one neuron to the other neuron, okay? So just talking a little bit more about synapse, the transmission and the action potential. So when the action potential is triggered, this causes information to be transmitted from one neuron to the other. Okay, it starts off by an electrical message, but it then changes into a chemical message, because remember, it's neurotransmitters that are chemicals. So ultimately is the chemicals we need to get from one neuron to the other. So it has to change into a chemical message as it reaches the end of the neuron. Okay, so the gap, remember, between the two neurons is called a synapse, or the synaptic cleft. When the presynaptic neuron, the first neuron, is stimulated by an action potential, it releases all of its neurotransmitters into the gap, into the synapse. What these neurotransmitters do, obviously, they are released into the synapse, and they're trying to bind to the receptor sites on the next neuron, which is called the postsynaptic neuron. When this happens, the calcium channels open. So remember we talked about the nodes of Ranvier. Yeah, the calcium channels open and the calcium ions come flooding into the neuron. Okay, the neurotransmitters are then released into the synapse, into the gap, and they obviously try to attach themselves to the receptors of the postsynaptic neuron. Now, sometimes reuptake can occur as well. So reuptake is when some neurotransmitters do not bind to receptors. So what happens is the presynaptic neuron will take them back up again. They'll obviously be used up again. So this diagram really shows example of synaptic transmission. So we've got the presynaptic neuron here, and we've got the postsynaptic neuron there. So what happens is those neurotransmitters, as the action potential is triggered from the other end of the neuron, okay, that will cause the neurotransmitters to come to the sort of end of that neuron there, the axon terminals. And those neurotransmitters, those vesicles there with neurotransmitters in them, are then released into the synapse here. Okay, so this gap is called the synapse. When those neurotransmitters are then released into the synapse, they will try to attach themselves to the receptors onto the postsynaptic neuron. Okay, so when they then attach to the receptors of the other neuron, then obviously an action potential is triggered. Okay, and they will obviously move on to the next neuron. Okay. Talking now about the way drugs affects neurons transmission. So it's very similar to what we just talked about. In the brain, there is something called a reward pathway, which is experience of a pleasant feeling. So this positive feeling leads us to repeat the behavior that activates that pathway, okay, making us want to do it again. For example, when you eat something that you really, really like, at McDonald's or your favorite food, you generally feel really good after you've eaten it, okay? And the reason for that is that it is stimulating or triggering your reward pathway, okay? So the diagram here, you can see there are different reward pathways for different neurotransmitters. So you can see dopamine's pathway is actually in blue, highlighted here. So it could go that way. Whereas serotonin pathway is a little bit different, so it could go that way around. Okay, so there are different reward pathways in the brain which different neurotransmitters and drugs actually take. Recreational drugs. So recreational drugs are those that obviously are taken for personal enjoyment. They are often called psychoactive drugs, and because they change the way the brain works and functions, and they often affect mood and behavior. Some examples of recreational drugs are caffeine, nicotine, alcohol, cannabis, cocaine, and heroin. And you need to be able to describe some of these for your in your exam. So we're going to come back to some of these examples and have a look at how they actually affect behavior. So drugs affect the brain's reward pathway, which, when it's obviously activated, causes us to experience this pleasant and rewarding feeling, which is known as euphoria. So just going into a bit more detail about how drugs actually affects the brain. So talked earlier about eating your favorite food. Okay, when you eat your favorite food, certain neurotransmitters might be released in your brain. The reward pathway is activated and it makes us feel good. In the exact same way, the way drugs work is by hijacking the reward system in the brain. And what they do is produce the exact same pleasant feelings, but a little bit more intense. So neurotransmitters are chemicals in the brain. So examples we talked about earlier, dopamine and serotonin. And neurotransmitters and drugs work in the same way. They work by changing neurotransmitter levels, especially dopamine. Dopamine is implicated in a lot of different behavioral disorders, okay? Heroin increases dopamine at the synapses, and that increases the amount of dopamine in the reward pathway, which creates that euphoric feeling. The brain then naturally reduces dopamine production, which wears off slowly. So obviously after taking a recreational drug, it's obviously not going to have an effect forever. Okay, after the effect actually starts wearing off, okay, and becomes weaker, this will leave a slightly lower level of dopamine than originally that person had. Okay, so this causes dysphoria, which is the opposite of euphoria, which is unpleasant feeling of maybe feeling a bit low, feeling a bit depressed. And then what happens is this motivates the person to take more of the drug, heroin, to produce the exact same feeling of euphoria. And what happens is, as the person takes more and more of that drug, what happens is their dysphoria also increases. So after each drug cycle, their original levels of dopamine in the body will become lower and lower. Okay, and this is what leads to addiction and physical dependence, because some of the withdrawal symptoms experienced are dysphoria. So some examples of some of the drugs. Caffeine. Caffeine takes effect within minutes, so it blocks chemical signals in your brain and also blocks reuptake of dopamine, so it stops you from feeling sleepy. Moderate doses, caffeine does or has been known to improve mental ability, things like reaction time, memory, but again, there's very limited research on caffeine's effects. Nicotine works by mimicking the actions of acetylcholine, which is a naturally occurring chemical in the brain, and that obviously binds with the receptor molecules. It increases the amount and transmission of dopamine as well. So another one that affects dopamine directly. Amphetamines increase the level of two of the brain chemicals, noradrenaline and dopamine again, by reversing reuptake. So what it happens is it leaves more dopamine in the synapse, which causes that feel-good euphoric feeling. Cocaine exaggerates changes in noradrenaline and dopamine again, increasing alertness and causing that feel-good factor. And the way it works is it blocks the reuptake of dopamine in the presynaptic. So more dopamine actually stays in the synapse. So it's not being taken back up by the presynaptic neuron. And heroin increases the amount of dopamine in the brain by boosting the dopaminergic synapses and causing euphoria. What happens is, as that wears off or is regulated by the brain system, it causes a negative dysphoria feeling. So this obviously motivates the person to take more of the drug to reproduce that feeling of high, okay, which is experienced, and to stop them feeling bad. So these are some examples of drugs, obviously, that you need to learn for your exam. And really, we're going to be finishing off by looking at the evaluation, really, of some of the research in drugs. Some examples of studies here. So it's important to learn some supporting studies. We've got Weinshaker and Schroeder, 2007, use lesioning. So lesioning studies are examples where they will cut into the brain or inject the brain with something. So these guys looked at lesion studies of the mesocortical limbic pathway in mice and found that neurons in this region were unable to produce dopamine, which is obviously normally associated with rewards. This does not, however, occur in other parts of the brain. So what it suggests is that cocaine's effects are due to the activity of dopamine in the brain's reward system. So it's a supporting study because it shows that it obviously does affect the brain's reward system. There are, however, problems because this obviously study was carried out on mice, which a lot of studies in psychology, especially in the biological drugs topic, are carried out on animals. However, the problem with animals is although the basic transmission process of drugs in animals and humans is exactly the same, the human brain is a lot more complex than the brain of a rat, so it's going to be different. A study by Nora Volkow conducted PET scans. So brain scans were carried on humans to track dopamine transporters in cocaine-induced participants. They found that as more dopamine transporters were occupied by dopamine, subjective experience also intensified. So that just shows that similar findings are found in humans and animal studies, really. And really, as knowledge of the effects of drugs increases, and research has always been carried out on drugs and their effects on the brain and behavior. So as we're learning more and more, treatments for drug addictions are becoming more widely available. For example, drugs such as naloxone can now help people to manage withdrawal and reduce symptoms, and can be used for people that have addictions to help them. A study by Olds and Milner in 1954 found that when stimulating areas of the dopamine pathway, rats would continue to press a lever due to the rewarding effect of the stimulation. So again, another supporting study there. Okay, so really that just concludes the session really on central nervous system and how drugs can affect the body.